In this video, we are going to do regulation of eukaryotic gene expression. And in this, I have made a schematic, kind of like a flow chart to enlist different types. First is the coordinate regulation. So we call it A. And then we have the mRNA processing and use step. And then through variations in DNA, we can regulate gene expression. And further, its headings are going to be the glucose circuit first, the hormone response system, and under the hormone response system we have the intracellular receptors and cell surface receptors. In the mRNA processing and use, we have the alternate splicing, alternate polyadenylation, mRNA editing, and mRNA stability. And under mRNA stability, we have iron metabolism and RNA interference, and also finally mRNA translation. And in variations in DNA, we have access to DNA, amount of DNA, arrangement of DNA, and mobile DNA elements. So the first is the galactose circuit. So in this, in coordinate regulation, so honestly, in coordinate regulation, the things that we look at is the special transcription factors. So we look at the STFs, special transcription factors. So they have two domains. One is the DNA binding domain, so we call it the DNA binding domain, and the other is the transcription activation domain. This transcription activation domain is going to recruit co-activators such as HAT and general transcription factors, right? We already went through on this topic before in the previous video. Now we had galactose circuit. Now in galactose circuit, this is basically an example to how coordinate regulation occurs. Now galactose circuits in yeast, we're going to take the example of yeast. In yeast, when there is less glucose, the cell of the yeast, what it does is that it increases the genes required for proteins that cause metabolism of galactose. So that is the basic concept that you need to know before going in how this works. Now we need to see how these genes are activated to metabolize galactose. For this, we have first is circuit off, when galactose is not being produced, or metabolized, right? So this is our target gene right here. This is the target gene that I drew in front of you. And what happens when in this we have to the gene should be inactivated. When the gene is inactivated, we have, this is our STF, the special transcription factor right here. Let me color it good and nice. And this is called as GAL4. This is the GAL4 is the STF. So this is and it has a transcription activation domain above and a DNA binding domain below, right? So with DNA binding domain, it is attached to the DNA, and then we have the transcription activation domain. Now when the circuit is off, what we need is to stop or to cover this transcription activation domain. So we do this by a protein that is called as the GAL80. So GAL80 is going to cover the transcription activation domain with GAL4, and that is how there will be no transcription. Okay. But what happens when the circuit is on? When the circuit is on. So I'll make a good DNA here. This is the DNA. We have the target gene. So what happens is, here we have our STF. This is our STF with its DBD and its DAD. What we need to do is to remove this gal-80. This gal-80 is removed by another protein that is called as gal-3. So gal-3 is going to remove by binding with gal-80. When this is removed, the transcription activation domain is going to open up, and now this is going to activate the gene and cause transcription. It is going to recruit GTF, and it is going to recruit the coactivators, and it is going to increase transcription. That is how a galactose circuit works. Okay, so next is the hormone response system, and the first of it is the intracellular receptor. In this, ladies and gents, we will take the example of hormones. The hormones which are lipid soluble, which means they can cross the plasma membrane, and these are steroid hormones. So the hormones that can cross the plasma membrane, they will have receptors which are intracellular. So that is always going to be the case. Now we will take the example of cortisol here. I'm going to change the color first. Let's do this good blue, no, maybe red, orange, or let's do orange. We have, when cortisol enters the cell, so this enters the cell, the cortisol, 2 molecules of cortisol are going to combine with 2 molecules of the glucocorticoid receptors, GR. So this is the glucocorticoid receptor, and this will form a complex which is a dimer. So it will form a dimer. Now this dimer, this is both our receptors. So this is the intracellular receptor binding with the cortisol, and it will form a dimer. Now this dimer is actually a special transcription factor. When this dimer is formed, its DNA binding domain will activate because it has the zinc fingers. It is now activated. Now this G dimer, which is basically now a special transcription factor, it will go inside the nucleus to the DNA. It will go to the DNA right here, and in the DNA it has, the DNA, there are certain regulatory, we have the glucocorticoid response elements. Now in these response elements, this dimer is going to bind. So I'm going to show you with orange. This dimer is going to bind here, and then of course when this the DNA binding domain is attached to the GRE, then it is also going to have a TAD that is going to activate coactivators, and those coactivators are going to increase the transcription of the target gene, and that will increase the different proteins that are will be transcribed. And that is it for this. Now the next is the cell surface receptors. Now these are for those hormones that cannot cross the lipid membrane, and they are going to have receptors, protein receptors on the cell surface. Now the example to this, you can take any hormone I'm going to take here, and this is our cell membrane, and this hormone is going to attach to its receptor, and this is going to cause the conversion of ATP to CAMP, that is the second messenger system, and this CAMP is going to cause phosphorylation of Crab. Crab is another protein. Now this Crab protein is now a STF. This is a special transcription factor. This Crab is going into the cytoplasm, in the nucleus, and in the nucleus it is going to get on the the CRE, the response element, that is the CRE. And then the CRE is going, this is the response element, Crab is going to get attached here, and it is going to activate via TAD, its transcription activation domain, and it is going to increase transcription. As simple as that. In the next video, we are going to look at messenger RNA processing and use and its role in gene expression regulation. Thank you so much.